Hello and welcome to Module 2, Lesson 1 of BIO100A regarding inheritance patterns and how to solve test crossing using Punnett squares. Genes are DNA sequences that carry information, and they are the units of inheritance. Each of us has two versions called alleles of every gene, one from mom, the other from dad. Through the process of sexual reproduction, these two alleles are separated into the gametes, the reproductive cells, and then they are combined during fertilization when sperm and egg meets to form a new being.
We wonder about many inherited traits that we may or may not pass down to our children. Who do I blame for my straight hair, or curly hair, or big feet? How did I get my father's eyes, and my uncle's chin, and my grandmother's smile? Hearing a new baby described as having his father's nose and his grandmother's lips could make someone think we're all cobbled together out of a bunch of used parts. Where do we begin and our parents end? Let's recall from Chapter 8 that sexually reproducing organisms carry two copies of every chromosome in every cell, except their sex cells. Humans, for example, have 23 pairs of chromosomes, 46 individual chromosomes. At each location, referred to as a locus, on the two chromosomes of a pair is the same gene, one copy from the mother and one from the father. As we noted in Chapter 6, each of the two copies of the gene is called an allele. Physical appearances don't always exactly reflect the underlying genes. A normally pigmented individual may have two copies of the pigment-producing allele, or they may have only one copy. In either case, the individual will look the same. The outward appearance of an individual is called their phenotype. A phenotype includes features visible to the naked eye such as coloration, height, or the presence of antlers. A phenotype also includes less easily visible characteristics such as the chemicals that an individual produces to clot blood or digest lactose. An individual's phenotype even includes the behaviors it exhibits. Underlying the phenotype is the genotype. This is an organism's genetic composition. We usually speak of an individual's genotype in reference to a particular trait. For example, an individual's genotype might be described as homozygous for the recessive allele for albinism. Another individual's genotype for the melanin gene might be described as heterozygous. Occasionally, the word genotype is also used as a way of referring to all of the genes that the individual carries. How do we analyze and predict the outcome of crosses? First, we must assign symbols to represent the different variants of a gene. Generally, we use an uppercase letter for the dominant allele and lowercase for the recessive allele. We represent the albino individual as lowercase m because it must carry two copies of the recessive allele m. An individual that is pigmented must be uppercase M, or uppercase M, lowercase m. If we don't know which of the two possible genotypes the pigmented individual is, we can write uppercase M underscore, where the underscore is a placeholder for the unknown second allele whose identity we aren't certain of. In the figure, we illustrate the cross between a true breeding pigmented individual, uppercase M, and an albino, lowercase m. Along the top of the Punnett square, we list individually the two alleles that one of the parents produces, and along the left side of the square, we list the two alleles that the other parent produces. We split up an individual's two alleles in this way because only one of the alleles is contained in each sperm or egg cell that it produces. The two gametes that come together at fertilization produce the genotype of the offspring. In the four cells of the Punnett square, we enter the genotypes of all the possible offspring resulting from our cross. Each cell contains one allele given at the head of the column and one allele at the left of the row. In cross 1, illustrated in the figure, every possible offspring would be heterozygous and would be normally pigmented, because all offspring receive a dominant allele from the pigmented parent and a recessive allele from the albino parent. In the second half of the figure, cross 2, we trace the cross between two heterozygous individuals. Note that each parent produces two kinds of gametes, one with the dominant allele and one with the recessive allele. This cross has three possible outcomes. 1/4 of the time the offspring will be homozygous dominant, both uppercase M. 1/4 of the time the offspring will be homozygous recessive, both lowercase m, and the remaining half of the time the offspring will be heterozygous, uppercase M, lowercase m. Phenotypically, 3/4 of the offspring will be normally pigmented and 1/4 will be albino.
So what did you learn today? You learned about genotype and phenotype. Genotype is the information in the genes, which is expressed or symbolized with letters, and phenotype is how these genes manifest, which we describe with words. You also learn about alleles, which are the versions of genes, and gametes, which are reproductive cells, that in that process alleles get separated and you're going to have only one allele per gamete. And then you also became familiar with Punnett squares, which is a simple tool to predict test crosses by putting the potential gametes on either side of the square and figuring out the different combinations. Thank you.